Good morning, everyone. Welcome to the January 2026 Zoom Citrus Seminar. Thank you for joining us. First, I would like to thank Deborah for her help and cooperation at our Henry County Extension Office in La Belle. Today's program offers one CEU for pesticide license renewal and one CEU for certified crop advisors. If you need CEUs, email me your name, email address, and license number. The title of the seminar presentation today is Development and Integration of Practical Tools to Manage HLB in Citrus Groves. Our guest speaker is Dr. Ozgur Batuman, Associate Professor in Citrus Pathology at the UF-IFAS Southwest Florida Research and Education Center in Immokalee. Because there is no silver bullet for HLB, the available tools must be integrated into regional integrated pest management IPM strategies and further developed in order to achieve disease control and maintain sustainable citrus production in the state of Florida. The intent of this talk is to highlight the tools that can be utilized for HLB IPM, many of which were developed by the research team at the Southwest Florida Research and Education Center in Immokalee. This includes the use of individual protective covers, IPCs, and plant defense inducers as preventative measures to protect young trees and new flush, as well as OTC trunk injections on mature trees to manage HLB under endemic conditions. Additional tools in the development pipeline, including an automated delivery system for ATC trunk injections, which could help reduce labor costs, will be discussed as well. Dr. Batuman, it's all yours. Please go ahead.
Great. Well, first of all, thank you very much, Monji and Deborah, for the logistic and giving me this opportunity to share what we are doing in Immokalee with, of course, with the help of a number of fantastic other faculty in IFAS throughout the states. So Monji started this presentation with the one sentence that I will probably keep repeating it several times, so sorry for that. And I'm a strong believer of integrated pest management strategy. As a plant pathologist, I know that some microbes can be very, very smart, smarter than us. So I will repeat that there is no silver bullet for HLB, especially such a challenging disease, right? And I will try to show a couple of slides to, remind you what I mean by silver bullet, or why do we need integrated pest management for HLB control. I'm not going to go in detail to introduce you to HLB, but just remember that HLB is not a new disease. It's a 100-year-old disease, and still we don't have one solution to control it. And further slides, I will be talking about what we've done so far and what worked, what didn't. But as a IFAS citrus team, we decided that we start our presentation giving a take-home message, and this is exactly what I'm doing here. And this take-home message, as you read here, and it will give you some ideas or outline of my talk for today in my seminar. And again, no silver bullet for HLB control. And as Monji mentioned, individual protective covers and mesh bags that most of you in Florida now familiar and adopted this. It's really protecting the young, newly planted trees for sometimes, and they are very effective controlling ACPs and some other pests that I will talk briefly that can be helpful to reduce in citrus grows if it's used properly. And obviously, oxytetracycline is antibiotic that we are all familiar these days, and we are using with the trunk injection of infected trees with the hope that oxytetracycline will reduce the bacterial titer and will give our trees to chance to hold on to fruit that they are producing before the harvest, rather before the harvest dropping to the floor and become unavailable for you. And here also I will be talking about the plant defense inducers. Actually, these are a family of chemicals that we identified by collaborating with Bayer and screening their vast agrochemical library to find some practical alternative to oxytetracycline solution, practical solution for you that you can incorporate into the integrated pest management for HLB that we are all trying to build. And of course, oxytetracycline and PDIs, you will see in my slides, if it's used probably, they can help you. Nevertheless, like all antibiotics, oxytetracycline can degrade, or bacteria quickly can develop resistance if you heavily rely on only one, chemical, in this case, oxytetracycline. And then, of course, some of the new antibiotics that I will be talking about can cause phytotoxicity if it's applied and without the proper caution. So I will share overall my experiences working with this tools that we've been trying to develop. Okay, and I will of course emphasize that a regional integrated pest management approach is the best approach until we really find tolerant or resistant citrus cultivars that we can incorporate to integrated pest management. So this sentence probably suggests that integrated pest management is needed until we have resistant varieties, but it's not true. When you have resistant varieties, you still need to do integrated pest management to protect that tolerant or resistant new varieties for longer time. As I said, microbes are smart and they can overcome resistance. So how to control HLB? So far, as I mentioned, many practices that growers here in Florida and other part of the globe where citrus grown used, but unfortunately none of them was alone enough to control HLB. So I started my list here as a control 101. As a plant pathologist, we always remind our growers that insect transmitted diseases requires these three fundamental management practices. One, reduce the insect that transmits the disease. Second, remove the infected plants. In other words, remove the inoculum from your field or grow. And third, start again with the clean material. In Florida, we tried these three, but one, which is highlighted with red, failed miserably. Hence, we couldn't remove the inoculum sources. That's why we couldn't stop the spread of this insect and the disease in Florida. And then the rest of the list, as you can see, HLB disease control tools, they are highlighted with red, indicating either those practices failed, or still we are trying to understand if they are really effective in controlling HLB. And some of them are, as I mentioned, remove and destroy infected trees. We couldn't do it after such a long canker eradication program. Quarantine program that we established didn't stop the HLB. Some of the chemotherapies and nutrition management practices fell short. Thermotherapy, one of them failed, and some of the bactericide that we tried spraying didn't work, and some other antimicrobials, and many other snake oils. When I say snake oils, do not get me wrong. I don't mean that your products are snake oil, but the way that we use them probably was not effective to control the HLB. And recently you will be hearing a lot that we are focusing on next generation technologies, highly advancing and really in some areas very effective tools such as CRISPR, RNAi, or transgenic approaches even being discussed and developed for our citrus growers to stay in business in Florida. Nevertheless, in the end of the list, as you can see, psyllid vector control, there are many practices work very well for psyllid and still working for psyllid. So all I said this just to tell you that there is no single effective control strategy for HLB. In other words, there is no silver bullet. What is integrated pest management, or what is silver bullet? What am I talking about? So integrated pest management, probably most of you are familiar with this famous pyramid that shows a number of practices together used to control pathogen or insects. In short, we will call pests, right? So when they are used in combination, they become integrated pest management. However, if we rely only on one of these layers here, for example, only the chemical spray, that chemical spray literally become your silver bullet. And it will work for a short time, but I can guarantee you it will not last long. That's why we are trying to develop number of tools that you can integrate into the integrated pest management strategy where we can control HLB better. By the way, I see in the chat something is written. I'm not following. Hopefully, Monji is following your questions or inquiries. Please feel free to unmute yourself and stop me if I'm going too fast or something is not clear. I will be happy to, try to clarify what I'm saying. In the end, happy to answer your questions. Okay. So again, integrated pest management, what I say is exactly using number of tools to control pathogens or insects together in our grows so that we can produce high quality fruits. So my program in SWFREC develops exactly the practical tools that we think growers can integrate into the IPM for HLB. So, but most importantly, I have to emphasize this, I do almost nothing alone. So I do collaborate with you guys, with industry, cooperator partners, and as well as many other great scientists, expert in the subject to develop number of tools that we are focused on. And the list over here briefly tells you some of them that I will be talking anyway. For example, we are always interested in actually in progress screening for new therapeutics, adopting new tools such as individual pest, individual protective cover (IPCs). And then automated trunk injection system, briefly, that I will be talking about with the hope that automation of trunk injection will lower the cost and will help growers to do the oxytetracycline injection more efficiently because of number of reasons that I will be talking a little later. And of course, we are also using resistant citrus as an interstock. Today, I will not be talking about And we are testing HLB resistant citrus cultivars that develop, by many excellent citrus breeders. And we are also collaborating with experts to develop RNAi CRISPR-Cas strategies or transgenic citrus trees that can someday will become available for you to use in the field. And I'm a virology expert, so I'm always interested in viruses. In that regard, we also look for some of the insect specific viruses that ACPs might have that can be used as a tool to control ACPs. And this is in progress, but I don't plan to talk about it today. And then, of course, we are always searching for effective new IPM tools. I will start with IPCs. And as you know, IPCs is not a new idea, or it wasn't our brilliant ideas. As basically, the industry in Florida was smart enough to propose this idea. And Monji and IFAS faculty, said, why not? Let's try and see if it's really effective protecting the young plantings from ACP feeding and HLB infection. And long story short, you now all know that it can be used in new planting and the reset situation, although it's costly and laborious, but can protect your plants, newly planted healthy plants, 2-3 years, depending on cultivar, from the HLB disease. And it is really very effective, excluding the insect. And here is one short slide to show you that we already published this, and this graph summarizes it. When plants, new citrus plants, are planted in the grow and put IPC on them, as you can see with the black dots here, indicating that up to 30 months until IPC is removal, they all stay clean of HLB. Whereas those plants that didn't get IPCs, 2 months, starting at 2 months, start getting infected with the CLAS and become affected by HLB. But as soon as you remove the IPCs, as you can see, the trend was similar, as if like you didn't put anything. They become quickly infected with CLAS. Okay, so IPC, if it's used properly, can protect your plants for some times, depending on cultivars. Of course, we also were interested to see if IPCs can protect our new small seedlings from other diseases, and we looked for other diseases such as citrus canker, greasy spot, and some other insects. As you can see, those that were covered with IPCs were taken and inspected and monitored regularly for other diseases, whereas along with that, those plants that were not covered with IPCs similarly monitored for some time. In short, for example, IPC covered plants, we noticed that citrus canker situation in all this time that plants were under protective covers did not have much citrus canker, indicating that IPCs also helped reducing the canker incidence. Nevertheless, greasy spot was slightly increased in the plants, on the plants that were in the IPCs, covered with IPCs, indicating the microclimate that we are creating can, be helpful for other diseases such as greasy spot to thrive. And also sooty mold that developed in some of that covered trees, indicating that the other practices such as spraying and controlling other diseases is still necessary. Again, it emphasizes integrated pest management, right? So it's only putting IPC is not solving your problem, so it means you have to do other things. One thing that I would like to remind you, of course, this is happening in our trial, your grows, everywhere. But when shoots such as here, as it's shown, or the suckers coming out of the bag, this defeats the purpose. Okay, so this, when it's infected from here, entire plant is infected. So it's mean you wasted your time and money by putting this IPC. So it's mean we have to really remove the suckers, or we have to keep the branches under the intake, the IPCs all time. In summary, yes, IPCs are working, and so depending on cultivars, up to 3 years can protect your trees from getting infected, and some other diseases can be, lowered if it's properly used. Nevertheless, you need to do some additional practices to control other insects. Okay, and then of course we should not keep any green parts outside. And as a virologist and plant pathologist, that working with the phloem-restricted pathogens such as Honglongbing causing or associated bacterium, Candidatus liberivector asiaticus, we call CLAS, right? Phloem limited, so it's very difficult to send chemical materials to kill this bacterium or other phloem limited pathogen in the phloem. Hence, we focused on how to deliver some chemicals into the phloem in the right time and the right amount so that we may kill or reduce bacterial population in the citrus trees. Again, trunk injection is not our ideas. It's been practiced hundreds of years in other cropping systems, but we truly believe that if we want to tackle phloem-limited pathogen, we definitely need to study and learn how to deliver chemicals or therapeutics into the citrus phloem. Hence, we built a team. Long story short, we studied how citrus vasculature works, when is a good time to drill a hole and inject things, what type of things you can inject and where it goes, how efficiently distributes in plants, etc., etc. Long story short, now entire Florida has adopted this trunk injection idea, especially relying on oxytetracycline. In our trials and other researchers' trial and growers' trials, we are all convinced that if oxytetracycline properly used, it can help decreasing bacterial titers, improve overall tree health, and increase the fruit's yield, size, and internal quality. Okay, and of course, it's reduced premature fruit drop as well. So it's clearly in many places that oxytetracycline injection practice, it's working. One picture that it strikes me all the time whenever I look at it, you can see here the Duncan grapefruits in our SWFREC experimental grow. One on the left is not injected, right is injected. It's day and night difference in appearance of the tree. So it's clearly oxytetracycline trunk injection, when it's used appropriately, it can help. Of course, there are a number of challenges associated with OTC trunk injection. For example, we all agree that it's a labor intensive and it's costly practice. And in some instances, when it's used in the wrong amount and wrong time, can be phytotoxic and prone to degradation, like all antibiotics, right? When antibiotics exposed to the light and heat, they degrade very quickly, and oxytetracycline is not an exception. So we have to be careful, and I will try to show in further slides that this is the case for oxytetracycline too. And of course, as I said, as a plant pathologist, I'm always worried because I know for a fact, and I try to prove that sooner or later, pathogens can develop resistance to antibiotics. Okay, so to tackle some of the challenges, to help our growers to be, ahead of the game, we thought, okay, trunk injection, but can we automate it? And in our team, we had an engineer in Immokalee, Yanis Ampazidis, has done great job. Basically, we developed, ATIS, automated trunk injection system, where as if like robotic arm that in this case mounted on small ATV here will go out and grab the citrus trunk from both sides and inject the oxytetracycline from both sides. And indeed, our prototype worked quite nicely. Let me see if I can turn on my video. Yes, it's going. As you can see, the end effector at the end of the robotic arm clamped to the roots, the trunk of the citrus trees, now injecting, as you can see, the oxytetracycline here going down, and there is no pressure building up. It's mean there is no resistance here. In bottom line, we were able to inject 100 milliliter less than 30 seconds from both sides of the trunk. So it's mean this prototype indicating that it's doable, and still we are interested to hear from our growers or today, for example, attendees to share their feedback if such tool will be practical and useful for you so that we can continue to focus on this to fully automate it. I mentioned about antibiotics degradation, and as you can see here in this slides, when freshly prepared oxytetracycline exposed to the light and high temperature, this is a normal condition in Florida's growth condition here. When you go out, it's very sunny and hot temperatures. As you can see, oxytetracycline changes color from light yellow to dark brownish, reddish, orangish color. Gives feeling like it's something is happening. Maybe it's degrading, but is it really degrading by changing this color? We didn't know. So, but we and other growers asked this questions. Is it really degrading? And my graduate student, Lauren Fessler Matthews, exactly asked this question, and look at the oxytetracycline that was freshly prepared and exposed in different times duration to high light and temperature. And as you can see, by using the HPLC, a sensitive technique to quantify OTC, for instance, the amount of oxytetracycline in solution with the extended exposure times reduced quite a bit. And when she used this exposed oxytetracycline along with the freshly prepared oxytetracycline to inject trees outside in the grove, what we found out was not a pleasant results. Only freshly prepared oxytetracycline provided some bacterial reduction in injected trees, whereas those that expose up to 6 hours, I think, and more, did not provide any efficiency against sea lice population in the plant. In bottom lines, from this, we understand that it's only freshly prepared oxytetracycline should be injected, and I think when it's not degraded, we will have some efficacy. The similar question we ask also in laboratory setting. Of course, HLB bacterium unculturable, so we use proxybacterium, in this case, culturable bacterium, which causes citrus canker. We are all familiar, so we can grow in the lab and do all sorts of experiments, right? But before going there, some of the growers actually came with a very interesting idea by mixing oxytetracycline with ascorbic acid, AA, or vitamin C. They noticed that the color was not changing. And indeed, CRDF asked us to see if there is any truth on that. And as you can see here, oxytetracycline freshly prepared and exposed, the number of hours indicated here to the light and temperature, 86 Fahrenheit degree here for this much. As you can see, gradually the color changed to the brown, whereas when you are mixed with the AA, the color change was not happening. So it looked like, adding ascorbic acid to oxytetracycline slows the color change, but doesn't mean that it's slowing the degradation. Before that, to answer that, I also wanted to show you that mixing ascorbic acid with oxytetracycline, first of all, didn't prevent oxytetracycline to do job, at least to the bacteria that causing citrus canker in the in vitro assay. Here in the plate, as you can see, when you put oxytetracycline on filter paper and put on the bacteria growing medium, so oxytetracycline will kill the bacterium around it, and we will call this zone of inhibition where bacteria cannot grow. Hence, we can say that oxytetracycline is effective controlling this bacteria. And when you compare oxytetracycline alone mixed with the AA, as you can see, the zone of inhibition happened. It means AA did not interfere with oxytetracycline doing the job. So that was a fantastic news for us. Whereas AA only, as you can see here, there is no zone of inhibition. It means AA only cannot control the bacterium by itself. So what happens when you mix AA with oxytetracycline and go and expose them for a long time to the light and heat and inject to the citrus trees? As you can see in the graph, hopefully you can see that oxytetracycline that freshly prepared with same amount of AA did not prevent oxytetracycline's degradation in vitro assays. As you can see, for a while, oxytetracycline with or without AA was able to stop the bacterial growth. By the way, let me remind you, oxytetracycline is an antibiotic that is not bactericidal. It means it doesn't kill the bacteria, right? It's the bacteriostatic, so it stops the growth of bacteria. And as you can see here, for up to 3 hours, oxytetracycline with or without AA was able to stop the growth of citrus canker-causing bacterium, whereas after 24 hours, bacteria start growing again with or without oxytetracycline. It's mean oxytetracycline after 24 hours definitely degrading and losing its potency, and AA is not helping. Okay? So again, we are worried about the relying only on oxytetracycline, and our goal is to maintain an OTC's efficacy for a long time until more, sustainable solution comes along for our growers. That's why we are looking for rotational or alternating partners for oxytetracycline. To do that, we look at the other agricultural production system around the world, and we brought some of the antibiotics listed here, for example, and tested against HLB control. In other words, their efficacy on killing or stopping the bacterial population that causing HLB in the citrus trees. And we focused these because they were, first of all, approved for use in trees or other crops in the US or other part of the world, and they were easily soluble and injectable. Okay, so that's why we focused on this antibiotics listed here. And of course, we did not rely only on antibiotics, but we are also interested in plant defense inducer and other chemicals that can, with combination with oxytetracycline, can be synergistic and help reducing the sea lice population. In this case, PDIs, of course, proprietary material from Bayer. Regalia is a commercial product. It's a biological product. Actigard is well-known plant defense inducer, hence we use them. And injectable, imidacloprid, it's called Oxytet, was used in conjunction with oxytetracycline injection. I said it's a fact that sooner or later pathogens develop resistance to antibiotics. As you can see here in this slide, everything that is green It's mean a bacteria, a bacterium or bacteria develop resistance to streptomycin, not in the United States only, but in other countries as well. You can see the number of green boxes here, okay? Because some of the bacteria start developing resistance to streptomycin, kasugamycin was introduced to the agricultural practices. And as you can see, the blue boxes indicating that bacteria develop resistance to kasugamycin as well. And do you see anything orange in the slide? Orange box. Yes, that's the bacterium that develop resistance to oxytetracycline. Okay, so this is for fact that bacteria can develop resistance to antibiotics. So that's why we have to definitely find alternative or rotational product to OTC. I will give very short results from ongoing field trials about the antibiotics that we tested. First of all, I can tell you that none of the antibiotics that we tested performed better than oxytetracycline. However, some oxytetracycline rotational products that was either injected first in the fall or summer before oxytetracycline injection or after oxytetracycline injection provided some very nice control. But that, treatment has to be repeated again. And indeed, last Monday, we harvested a Hamlin plot that were treated with this OTC rotational crop antibiotics. So when the data becomes available, I will be happy to share with you by writing short citrus industry magazine articles or some other ways to share with you our results. But I can tell you that we need to repeat these experiments before definitely saying what is working, what is not working, okay? And I should caution you if you are interested to use this antibiotic that they can cause phytotoxicity. As you can see here in excised leaf assays, in the lab condition, in the greenhouse, small plants injected with different type of antibiotics, they can all cause severe phytotoxicity. So please, when you are trying some antibiotics, be careful that you can hurt your trees. Especially when it's young trees, I recommend not injecting anything. Even in the field, some, including here, I'm showing this is oxytetracycline. Actually, as you can focus on here, there are some branches in the whole tree that it looks like more yellowish and golden color, indicating that some sort of phytotoxicity because unhomogeneous distribution of oxytetracycline, indicating that it's focused or mainly went to this branch and caused little phytotoxicity. Of course, this plant bounced back from this, symptoms, but nevertheless, again, you have to be careful that some antibiotics can cause phytotoxicity. Here, some phytotoxicity in the trunk-injected trees. The trunk split is also possible with other antibiotics. Now, I have a very limited time left, so I'm going to go faster, but I'm switching gear here to talk about plant defense inducer. So the idea with the plant defense inducer is to protect the young developing shoots. As you can see here, this is the experimental design. The idea is spraying this plant defense inducer very young shoots and try to protect this as they develop and become inaccessible to ACPs. And as you know, psyllids love small, young, developing flushes. That's why you have to protect this young flushes. In this experiment, screening for Bayer's chemical library, we developed a method where we can spray this healthy plants, please pay attention, young healthy plants in the greenhouse, to protect these flushes, 3 times sprayed. We call it priming. It's mean activating the natural plant defense in this young healthy plants. And then we challenge inoculate it with the psyllids that carrying CLAS number of times. And we continue to monthly spray, protect this developing flushes for a year or more, and monitor the bacterial buildup in this plant. Here is the slide from the experimentation, how we challenge this sprayed plants. As you can see, we introduced 5 female and 5 male ACPs in each bag, and we continuously challenge inoculate these healthy plants as long as their young flushes were developing. So long story short, water-treated plants, as you can see here with the blue line, when they are challenge inoculated, as soon as 2.5 months later become infected, and bacterial titer in these plants become very high as short as 4 months. But when you compare with one of the PDIs that we tried here in this experiment particularly, this plant didn't get infected up to 6 months. We couldn't detect the CLAS in there. And then the bacterial buildup was very slow. So in other words, this plant defense inducer significantly delayed the infection and reduced the bacterial buildup in healthy protected plants. Then we quickly said, well, now we have oxytetracycline that we are injecting outside in the field. Can we reduce the bacterial population in these heavily infected trees and protect the young flushes that will come afterwards? And we were very naive to think that oxytetracycline injection or CLAS reduction will synchronize the shoot formation, and we will be able to protect those shoots. But nevertheless, we tried these PDIs in the field condition. Long story short, only oxytetracycline with or without PDIs was effective enough to reduce the bacterial titer, whereas only PDIs, or for example, brassionosteroid also was included here, was not enough to reduce bacterial titer this in heavily infected trees that grown in the fields. These are 7 8 years old Valencia trees. And when we look at the Valencia plot, for example, fruit drop, there was no significant difference between water injected, non-injected, or other OTC injected than PDI sprayed plants. There was no difference. Nevertheless, OTC injected plants had some increase in the yield, but we did not find this yield increase in the PDI sprayed plants alone. Why PDIs didn't work outside? I know I don't have a lot of time, but I just wanted to tell you, for example, over here, if you look at this disease resistance plant, how, strong jasminic defense. This is the immune response to the CLAS happens. For example, Australian lime, right? When CLAS are introduced by the psyllids, they secrete a signal that can very quickly recognize with the machinery and triggers the jasminic acid dependent defense system, right? This happens regularly in the disease resistant citrus cultivars. But this cannot happen in susceptible cultivars that we have, like Valencia, Hamlin, all other varieties that we have, because of faulty recognition, okay? So there is no strong jasminic acid defense system happening in susceptible varieties. And also we know that there are molecules that play pivotal role balancing salicylic acid and jasminic acid defense pathways. And if that balance thrown away, it is very difficult to build back. That's why, remember, that PDIs that we use in the greenhouse on healthy plants did magical work, but did not work in the field because the mechanism plant defense mechanism was severely impacted in outside after such a long time of a CLAS infection. Okay, so in other words, I'm summarizing here, the balance was thrown away in heavily infected plants or mature trees. So it's very difficult to build that balance back by only relying on plant defense inducer spraying. However, and I will connect this soon, when imagine that you are moving the IPCs and your plants are healthy and you have young flushes, there you can start using plant defense inducer in conjunction with the insecticide to protect those young flushes for number of years until they become eventually infected and then can be injected with oxytetracycline, right? But before jumping to that ahead of myself, I just wanted to show you that plant defense inducer can also, control other pathogens because you are triggering a defense that can be effective on number of insects and pathogen. In this case, the plant defense inducers that we use was very effective reducing the number of the citrus canker lesions on inoculated leaves. For example, here, as you can see, non-treated plants had many lesions on inoculated areas, whereas Actigard, commercially available plant defense inducers, or Bayer's product here did have very little canker lesion, and it was comparable to copper spray, right? Copper we are all familiar with. And here is the close-up picture to show you that this is non-treated negative control. In injected areas, you can see many tiny bacterial lesions, whereas PDI, for example, here, PDI 31 or PDI 26 sprayed plants did not allow bacterial canker to develop. I will quickly pass this because I don't have a lot of time and I want to leave a couple minutes for your questions. I want to reiterate what I was just saying a couple minutes ago. So some of the tools that we really focus actually can be practical if it's used properly in proper time and way. For example, I hope we are all in agreement that IPCs can be used at planting and protect our trees for up to 3 years. Of course, along the way, maybe you have to switch to the larger IPCs. Then there is an economy component comes in that you have to think about it. But then you can protect these plants until start bearing fruit. But however, when you remove the IPCs and if you do nothing, they will be infected less than 2 months. Okay? And so it means you have to definitely continue to control Asian citrus psyllids and continue to properly provide irrigation and nutrition to this plant. But at the same time, you can also protect this young flushes from reinfection by the psyllid feeding by using PDIs or strong insecticide regimen to protect those young flushes as long as you can. Okay, but when you start seeing the symptoms on this plants, it's time to think about injecting oxytetracycline. Of course, we are injecting oxytetracycline manually in these days, but hopefully later in the future, maybe we can have an automated machine that you can integrate to our integrated pest management strategies. In the meantime, we will continue to, evaluate for new therapeutics. We will look at the resistant interstock ideas, RNAi, transgenic, and some biological controls using insect-specific viruses, and whatever we can find to bring it up for you to integrate your management practices. In conclusion, I will repeat myself. There will be no silver bullet for HLB control. Okay, so we have to think integrated pest management. And with the proposed IPM tools that I just showed, we can stay productive And I know and I acknowledge that it's not cheap. Unfortunately, producing juice is not cheap any longer. But of course, we definitely need HLB tolerant and resistant varieties and integrate these varieties as soon as possible to our integrated pest management tools that we are using these days. And I still believe, and I will repeat this, that fighting along to HLB it's not going to be effective. We have to come together and fight the HLB with integrated pest management that is implemented regionally so that can be more effective and more sustainable and more long-living strategy. And of course, we definitely need resistant varieties yesterday. So with that, first of all, I would like to acknowledge your attention and participation in this seminar. I really appreciated leaving your work and coming to hear me to hear what I got to say. But at the same time, I have to definitely acknowledge my team and the lab members that did all the hard work. I did nothing. But also, I have to acknowledge all growers that helped me with anything that I needed. I'm very grateful on their support. Of course, the funds coming from CRDF, Citrus Initiative, Bayer, and NIFA, etc., was very helpful to do all this research that I gave you a brief description about. So with that, I will stop here and I will take questions. Thank you.
Thank you, Dr. Batuman. There are two questions from Dr. Castle, but I think you answered those questions.
Mr. Castle, you're unmuted.
I'm muted.
No, I can hear Monji. You are good, Monji. Can you repeat that question?
If a tolerant rootstock and cyan were available tomorrow, what would happen to IPM and how would the new plants be integrated into the industry?
Excellent question. Thank you very much, Dr. Castle. This is exactly I want to emphasize again. Having a tolerant rootstock, by the way, we have tolerant rootstocks. It's not really helping us, right? So by themselves, they are fantastic. But when you put susceptible cyan on it, story is changing. And with the resistant rootstock, it will probably be similar. Nevertheless, in ideal situation, let's say we have tolerant and resistant combination that is really reducing the disease severity. It will still require other practices. For example, you still need to reduce the psyllid population by insecticide. Okay, you will still need to give proper irrigation. You still need to give proper nutrition to protect that resistance that came available today to as long as we can. Because, as I said, pathogens know how to develop, how to mutate and develop resistance to everything. Okay? So, but of course, tolerant rootstocks, resistant rootstocks, science, and their combination is immediately needed for us to fight effectively to HLB today.
Another question. What effect does the IPM approach have on juice quality?
Well, every practice combination that we will put together, in the end, we have to look at the yield and the fruit quality. Okay? For example, for fact, I know that oxytetracycline injected, even oxytetracycline injected alone, trees' fruit, their yield increases fruit drop reduces, internal fruit quality, Brix increases significantly. So it's mean addition of IPM tools has to provide a return of investment. Otherwise, it is not suitable or sustainable practice to become the part of the integrated pest management. So it's a package that we are building that will help us to produce better, more fruit. I hope I answered the question.
Another question. Is OTC injection justified as an emergency tool? Can IPC alone sustain productivity long term?
My straight answer is no. First of all, I'm sad to say, as a plant pathologist, oxytetracycline is unfortunately only therapeutic so far that is working. Okay? So oxytetracycline actually giving a time for our industry to develop other more sustainable tool to stay in business. But relying only on oxytetracycline because of number of reasons that I talked today, it's not logical. And it will not, unfortunately, keep us in business if we only rely on oxytetracycline injection. That's why we need to develop other practices. And IPCs, speaking of IPC can be kept only for such a short time. Then you have to definitely remove that IPC or build cups structure like many of the entrepreneurs in Florida are doing it, and they are doing a great job. However, the investment is significant. So I'm not an economist, so I cannot comment on that. Switching from juice producing in the outside with the minimum input to switch to enormous input to produce juice in Florida is viable business. I cannot comment on that. But IPCs are sooner or later has to be removed. So we have to have another tool to prolong the health of those protected trees. So in this scenario, I am proposing to use insecticide to reduce the reinfection rates and plant defense inducers to continue to protect that developing tissues. And with the healthy plants, hormones are synced, we will continue to have synchronized shoot and root flushing. So it's much better control everything on that healthy growing plants than already severely infected mature trees that flush here and there. You cannot synchronize that. That's why most of the applications to reduce the psyllid populations or reduce the bacterial population is not effective, unfortunately, because of that unsynchronized growth in Florida condition.
Another part to that question, does trunk injection extend the productive life of all the trees?
All the trees, it's a stretch. There are some trees, I think it's better not to inject even. So again, this tool can only do so much, right? So when tree is severely dying back and is shutting itself and going to die, oxytetracycline alone, I don't think that it will be practical enough or economical enough. Okay? First of all. Second of all, oxytetracycline still it's not homogeneously distributed from one injection size, okay? So there are only one branch or two branches in the tree might get oxytetracycline and do something. What about the other part of the plants? That's why now we are focusing on injecting from both sides. But now the cost is increasing, right? So the practicality become question here. Okay, and remember, and many of the experienced audience here with the citrus diseases in Florida, we have a citrus blight disease. It didn't go away. It's still there. You cannot inject blighted trees. So it means we are trying to inject trees that is already blighted and oxytetracycline is not uptaking and moving. So it means we are already spending money for nothing to those trees. Okay? So we have to think about these factors when we are making decision if oxytetracycline is suitable practice for us. And obviously, I do not recommend injecting oxytetracycline to young trees right after, for example, IPC removal. First of all, oxytetracycline has no residual activity, okay? So the plant knows how to metabolize that thing. And if there is no bacteria when you inject oxytetracycline, that oxytetracycline is water to the plant. And actually, you are increasing the likelihood of developing resistance to oxytetracycline. So don't do that. First of all, trunk injection, young trees, it's traumatic experience for the young trees. You are injuring them. Second, antibiotics are phytotoxic, as you can see. And third, when there is no bacterium, oxytetracycline is doing nothing. All right. Our best bet is protect that healthy looking trees coming from IPCs with insecticide and plant defense inducers or other tools that other IFAS researchers are talking about. Okay. So please think about these combinations. And of course, you have to look at the economy behind this.
There are questions about the cost of OTC.
I will try to answer, but the best person to reach out is Tara Wade. She is a team member in SWFREC, and she did a survey, and she has interesting results. As far as I remember, the cost of oxytetracycline injection per tree, given the common practices we have, the way that we do it here in Florida these days, is about $1 a tree. Of course, the density of trees and amount of oxytetracycline that you are injecting, it can be factored, or how many times you are injecting. First of all, we can afford only one injection a year from one side of the trees.
We
For
will
example,
cover
in our
OTC.
trials, we know that there's a big difference injecting from one side only versus from two sides.
We will cover OTC in another seminar in a couple of months.
Perfect. I hope that can be more, useful information than what I say here.
The cost per tree is around $1.25 to $1.50 per tree. Any other questions? Again, if you are interested in CEUs, please send me an email and include your name, email address, and your license number. Any comment, any question? Thank you all. Thank you, Dabra.
Thank you very much again for all of you joining today. I will be happy to, answer your question through email, text message, phone call. I'm very approachable person. Most of you know we work together. So please do not hesitate. Reach out anytime. I will be more than happy to, work with you and try to answer your questions as best as I can.
Thank you all for your participation and see you next month.
Thank you.
Thank you.
Thank you.